Welcome to another video by myhematology.com where we make hematology simple. And today's video, we will explore the G6PD deficiency, glucose-6-phosphate dehydrogenase deficiency. G6PD deficiency is an inherited hemolytic anemia due to a red blood cell enzyme defect. Hemolytic anemias are defined as those anemias that result from an increase in the rate of red cell destruction with the release of hemoglobins into the plasma, not due to aging. G6PD deficiency is an X-linked defect whereby the gene G6 that encodes G6PD is found on the X chromosome. So in this case, in inheritance wise, they will affect the males, means that the males will display the symptoms while females are usually carriers of the disease. This disorder is one of the most common disorders worldwide. The degree of deficiency varies and is often mild in Black Africans, more severe in Orientals, and most severe in the Mediterraneans. For the optimal function of the red blood cells, we have the red blood cell metabolism pathways, and there are four major metabolism pathways involved. The first one is Embden-Meyerhof pathway, whose main function is to supply energy to the system, whereby they power the iron pumps in the red blood cell membrane. And then we have the methemoglobin reductase pathway, whose key action is to maintain the hemoglobin in a reduced state for oxygen delivery. And next, we have the hexose monophosphate shunt, whose key action is to reduce harmful oxidant in the red cells. And finally, we have the Lewer-Daring-Rapoport shunt, which produces 2,3-DPG to regulate oxygen release from the hemoglobin. Let's take a deeper look at how G6PD affects the hexose monophosphate shunt. G6PD is the catalyst in the rate-limiting first step of the pentose phosphate pathway, which uses glucose-6-phosphate to convert nicotinamide adenine dinucleotide phosphate NADP into its reduced form NADPH. In red blood cells, NADPH is critical in preventing damage to cellular structures caused by the oxygen-free radicals, for example, the hydrogen peroxide. It does this by serving as a substrate to the enzyme glutathione reductase. Reduced glutathione can be used to convert hydrogen peroxide to water and prevent damage to cellular structures, particularly the cell wall of the red blood cells, since they have limited capacity for repair once mature. So, in a normal condition, any extra oxidative stress can be handled by this system. However, in G6PD deficiency, when the red cell is challenged by an oxidative stress, for example, ingestion of mothballs, both lipids and red cell membrane proteins become oxidized, as there will be not enough reduced glutathione produced to convert the increased level of oxidants to water. And this will cause the hemoglobin to denature and precipitate intracellularly to become Heinz bodies. And the presence of these rigid inclusions causes the red cell to become trapped in the spleen, and sometimes what the splenic macrophages do is surgically incise the portion of the red cells that contains the Heinz bodies. In these circumstances, the red cells may escape with a gap appearing as bite cells, or these rigid and fragmented cells may also lead to intravascular hemolysis. A variety of infections and drugs can trigger acute hemolysis in G6PD deficient individuals. Ingestion of fava beans may trigger acute severe intravascular hemolysis as a condition known as favaism. The glycosides vicin and convicin in fava beans are responsible for hemolysis. They generate aglycons that octo-oxidize and produce free radicals. Other agents that may cause hemolytic anemia in G6PD deficiency includes mothballs, infections, and other acute illnesses, for example, diabetic ketoacidosis or oxidative drugs, for example, antimalarials, analgesics, and sulfonamides and sulfons. G6PD deficiency is usually asymptomatic unless triggered by an oxidant. There could be neonatal jaundice and also acute hemolytic anemia, which is caused by rapidly developing intravascular hemolysis with hemoglobinuria during the hemolytic crisis. The anemia may be self-limiting as new and young red cells are made with near normal enzyme levels. Between crises, the blood count is normal. However, in a crisis, the full blood count will have low hemoglobin level and reticulocytosis, and the peripheral blood film may show contracted and fragmented cells with white cells and blister cells, which have had Heinz bodies removed by the spleen. Specific tests include decreased fluorescence in the fluorescence spot test, and Heinz body may be seen in the reticulocyte preparation, particularly if the spleen is absent. DNA analysis will be positive for the gene mutation. Biochemical parameters will show increased serum bilirubin and lactate dehydrogenase levels, but reduced serum haptoglobin level. The treatment and management of G6PD crisis include removing the underlying cause. For example, drugs must be stopped for treatment. And there will be blood transfusion where necessary for severe anemia. Underlying infections must also be treated, and if indicated, splenectomy may be carried out. So let's have a recap on G6PD deficiency. G6PD deficiency is an X-linked disorder where the gene is found on the X chromosomes. And usually, this disorder is asymptomatic. And perhaps there could be neonatal jaundice or hyperbilirubinemia. But in acute hemolytic crisis, there could be hemoglobinuria. The causes of G6PD deficiency include favaism, or when there is ingestion of oxidative drugs, for example, antimalarials, antihelminthics, for example, mothballs, infections, or sulfonamides ingestion. In laboratory diagnosis for a crisis, there will be a reduction in the hemoglobin level and there will be reticulocytosis. The peripheral blood smear will indicate a moderate anisopoikilocytosis, polychromatic cells, or Heinz bodies in the supravital staining. There will be a decrease in the fluorescence spot test and serum haptoglobulin, and an increase in serum bilirubin and lactate dehydrogenase, and positive DNA analysis. Treatment and management of G6PD deficiency includes stopping the ingestion of particular drugs or beans, transfusion if necessary, treating the underlying infection, and splenectomy in rare chronic hereditary non-spherocytic hemolytic anemia. If you like this video, click like and subscribe for more videos, and thanks for watching.